Hello and welcome to today's webinar. My name is Kristen Turner. I am the Vice President of Global Sales Operations here at LKC, and it is my honor today to moderate our first presentation of our Women in Visual Electrophysiology webinar series. This webinar series actually stemmed from an idea from two of my wonderful female colleagues. We are all incredibly lucky to get to work with the most talented professionals in this field from all over the world. And we wanted to do something special that celebrated and honored specifically the many women that we get to work with. Today we welcome Dr. Delia Cabrera DeBuc from Bascom Palmer Eye Institute, and she will be presenting her research on using ERG as a biomarker for Alzheimer's and dementia. Thank you, Dr. DeBuc, for joining us. We're very happy to have you tonight. Before we dive a little deeper into Dr. Cabrera DeBuc's research, I thought it would be nice to start by asking her a few questions as it relates to her career in visual electrophysiology. So my first question for you is, how did you originally become interested in visual electrophysiology?
Yes, thank you for the nice introduction, Christine, and it's a pleasure actually to join you in this webinar as the first woman telling our colleagues what we are doing in research with electrophysiology. My first research activities were in the neuroscience field. Particularly, I developed models to identify epileptic sources using electroencephalography. So that was my first experience with the electrophysiology field, which actually allowed me to transition to visual electrophysiology once I initiated my research in the ophthalmic area.
Thank you. It's interesting to hear how you started in the neuroscience field and the path that you've taken to get to where you are now. My next question has to do with how would you like the research that you're doing to affect the world, and ultimately how visual electrophysiology is used in the world?
Yes, well, several lines of evidence suggest that measurements of the eye could be a simple and non-invasive way to detect early stages of Alzheimer's disease with high precision. Current tests to determine Alzheimer's are costly and invasive, and these are the tests that are conducted, with neuroimaging. Now, with research showing that Alzheimer's can start in the brain decades before symptoms appear, we're going to be able to detect and diagnose the disease in its earliest stages using, for example, low-cost and non-invasive eye tests, which could be actually a game changer for the field. Ideally, I think that a system that provides doctors electrophysiology biomarkers also offers a method by which non-invasive therapy can be planned, managed, and even delivered. So I think that physicians will appreciate the office-based low-cost tool that can yield, for example, sensitive measures to help patients see the value of a more individualized intervention option, and even offer an additional biomarker information to support differential diagnosis.
Thank you. I agree. Being able to use ERG to detect Alzheimer's earlier, or people who are at risks for Alzheimer's earlier than we can now, would be a game changer for the community, not just the patients, but also their loved ones, because this disease is truly heartbreaking for everyone involved. Yeah. Lastly, my final question would be, what would you recommend to a young person who's starting their career in this field?
Yeah, well, my advice for young researchers entering the field, for example, would be like, you should take risks and preserve your creativity at all costs. You need to cultivate your scientific curiosity, challenge critical thinking, and even enjoy friendly competition among your colleagues. I think it is important not to lose sight of why you choose to follow a career in electrophysiology in the first place, to find methods that can help to speed up treatment and even new discoveries.
Thank you. I think
You're very
that's
welcome.
great advice for any young person, really, and maybe some of us who aren't so young as well. A good reminder.
Yeah.
With that, I'd like to get into your presentation. One thing I wanted to remind our audience of, if you have any questions during the presentation, you can ask them at any time. You'll just submit them through the chat function on either your mobile phone or the desktop application. There should be a section on each labeled questions. So submit your questions throughout the entire webinar, and once Dr. Cabrera DeBuc is done, we will address the questions then. And with that, I am going to hand it over to you now, Dr. Cabrera.
All right, thank you. Good evening, everyone. It is my great pleasure to speak here today, and I would like to thank the LKC Technologies for their invitation and for facilitating this webinar. Today, I'm going to talk about how we can see the brain through the eyes, and specifically, I'm going to introduce biomarkers for Alzheimer's and dementia. Here are my disclosures. Our research is sponsored by the Finkel-Frankel Legacy Foundation, the Alzheimer's Association, and the National Institute on Aging. And this is the outline of our talk. Today I'm going to talk about Alzheimer's disease and eye biomarkers, and I will also briefly introduce the research we are conducting in our lab to identify multivariate biomarkers of cognitive impairment. There are more than 6 million Americans living with Alzheimer's disease. Unfortunately, access to get an early, accurate diagnosis is hampered by cost and convenience, and no real progress has been made in the fight against the disease since its classification more than 100 years ago. Diagnosis occurs late in the disease, and there has not been an effective way to detect the onset of Alzheimer's disease before it starts to appear. Besides, while current screening tests, such as the mini mental state examination, are reasonably accurate in detecting mild cognitive impairment, they are not sensitive to the earliest cognitive decline signs. These tests are also not designed to distinguish the most likely etiology. Also, neuropsychological cognitive assessments are highly time-consuming and require expertise in interpreting results, making their integration into existing standards of care infeasible. In addition, confirmatory biomarker testing with PET or cerebrospinal fluid testing is expensive, invasive, and time-consuming. Aside from finding a cure, the holy grail of dementia causing diseases, research is the discovery of biomarkers. However, the critical barriers to primary prevention of cognitive impairment in dementia-causing diseases are the lack of rapid, non-invasive, objective, sensitive, and low-cost biomarkers. Provided that previous studies have suggested that Alzheimer's disease initiates the case before it is clinically expressed, it will be possible to identify persons who will ultimately show the disease long before the early symptoms appear, as well as to target potential interventions to prevent Alzheimer's disease expression in such individuals at high risk. And this is why it's so important to identify good biomarkers of cognitive impairment. Now, visual disturbance is frequently an early complaint of Alzheimer's disease patients. Different studies, as you can see on the top of the slide, have described reduced visual performance on tests of contrast sensitivity, visual attention, motion perception, visual field, color vision, and also visual memory. It has also been reported that processing speed and visual short-term memory storage capacity correlated with cognitive measures in patients with mild cognitive impairment. This result particularly suggested that impaired visual processing could be a pathological feature present already at the mild cognitive impairment stage and might represent a different marker of upcoming Alzheimer's disease independently from memory deficits. These findings were compatible with the amyloid cascade hypothesis of Alzheimer's disease. So visual performance measures may have the potential to reduce the misdiagnosis rate in cognitive impairment at the earliest stage. Overall, these early studies pointed to the need to investigate the eye as a potential biomarker. And you can see that even at this early time, electrophysiology was used to investigate deficits in the visual abilities of the patients with Alzheimer's disease. Now, how we can see the brain through the eye? The eye is the only organ where one can examine directly the blood vessels that are part of the cardiovascular and the neurological system. The retina is a complex tissue at the back of the eye where the eyeball stores its rods and cones. Lots of delicate blood vessels, as you can see in this figure, and nerve cells that lead directly to the brain. The retina is a structure with multiple cellular layers, and the visual system is more than just the eye. It includes the retina, optic nerve, as well as the visual cerebral cortex in the brain and all interconnection. As the eye and brain share important structural and pathogenic pathways, we could see through the eye, and the conditions of the retinal morphology, circulation, and function could be used to assess the conditions of cerebrovascular and neurodegenerative diseases. Because the retina can indirectly reflect the microenvironment of the central nervous system, retinal imaging and visual performance features, as well as retinal circulation, could be investigated and used to see the brain through the eye. A major advantage here, compared to neuroimaging techniques like positron emission tomography, for example, is that imaging the retina is non-invasive and can be performed at low cost. Now, biomarkers are key when we try to detect early diseases in humans. For example, a biomarker is an objective measure that catches what is happening in a cell or an organism at a given moment. Biomarkers can serve as an early warning systems for your health. Particularly, pathological hallmarks of Alzheimer's disease include the presence of hyperphosphorylated tau and amyloid protein deposition. Currently, these pathological biomarkers are detected either through cerebrospinal fluid analysis, brain imaging, or post-mortem. However, these methods are not widely available because they are invasive, expensive, and not user-friendly. The eye possesses clear optics and shares many neural and vascular similarities to the brain, offering a direct window to cerebral pathology. There is an increasing research on developing eye biomarkers for Alzheimer's disease. You can see in this figure how many different features of the structure and function of the eye are currently investigated as potential biomarkers in Alzheimer's disease. Potential associations between the eye and brain in Alzheimer's disease have been investigated by many studies exploring the retina's structure and function. And here are some examples that I randomly selected. Notice the increasing trend in publications since 2011, when only 7 publications were in place. And during the last 2 years, 2019 and 2020, we have had 53 publications each year. Also, we have already 19 publications in 2021 around this particular area of research. Also, we have seen an increasing trend in the publications related to the use of electroencephalography in Alzheimer's. You can see that, for example, in 2011, only 1 publication was in place, and we have seen 7 publications last year. We also have increasing publications, as you can see, in electrophysiology research since 2011, as I showed you in the previous slide. And these are some examples of publications reporting studies on human subjects and animal models, all of them using electrophysiology and a combination of structure and function markers. Now, let me show you the retinal measures of biomarkers of Alzheimer's disease. First of all, we have that the amyloid beta-related impairment of synaptic transmission in the brain precedes disruption of neural network activity and induction of abnormal network synchronization. This trend reinforces the relevance of the search for the earliest electrophysiological Alzheimer's disease-associated alterations in the retina. As you can see in this figure, the retina contains complex networks of neuronal circuitry. This network can be divided into 3 layers of neuronal cell bodies and 2 layers of synapses. And such a spatial organization is optimized to provide the physiological output of the retina in the form of action potentials. These action potentials can be measured when the retina is stimulated with light using an electroencephalogram. The functional changes in the retina can be measured with several types of electroretinograms. We have the full field ERG, or the focal or the multifocal option, and the pattern ERG, and they differ basically in the type of stimulus that is presented to the eye and the lights that is used. We also have that the basic method is the full field flash ERG. In this method, the entire retina is stimulated in a full field GANs pulse screen, so all retinal cells are thereby simultaneously exposed to the light stimulus, and as a result, we measure the sum of action potentials. The bioelectrical response to this stimulus comprises a series of 4 component waves, as you can see here that we have the A, the B, the C, and the D waves, which are arising from different processes and locations in the retina. The A wave follows the onset of the stimulus and is generated by the photoreceptors. Immediately following the A wave is the B wave, arising from the bipolar and Muller cells of the inner retina. The C wave is generated by the retinal pigment epithelium due to an increase of the transepithelial potential by the light-induced bioelectrical activity of photoreceptors. So this wave gives us insights into the function of epithelial cells and photoreceptors. And the D wave indicates activity of the off bipolar cells. The amplitudes of the waves indicates the health of the part of the retina from which they originate. Another component of the electroretinogram are the time intervals. For example, we have the latency of response, which refers to the onset of the stimulus to the beginning of the A wave. And we also have the implicit time of the, that is a measure of the time interval from onset of the stimulus to the peak of the B wave. A variant of the full field flash ERG is a photopic negative response, which is a slow corneal negative potential arising after the B wave of the standard photopic ERG. The photopic negative response allows to measure the relationship between the function and the integrity of retinal ganglion cells. Now, we have different electrophysiological alterations in the Alzheimer's disease retina. For example, here you can see in this table studies that were reviewed from 1986 to 2017, and there is actually a good compound of studies, both using human subjects and animal models. I'm showing also specific results from selected papers from 2010, 2020, and 2021, summarizing what is happening in this particular field with electrophysiology. We have that in relation to human subject studies, the full field flash ERG has been studied in Alzheimer's disease patients under both scotopic and photopic conditions. The majority of initial studies that use this particular modality and also the pattern ERG did not detect significant differences between patients and age-matched normal subjects. However, recent studies using more advanced technology have reported alterations that are subjective of ganglion cell dysfunction with both the pattern ERG and the full field ERG, specifically with a photopic negative response, as you can see in this paper recently published in scientific reports by Dr. Asanat. In relation to animal models, a photopic full field flash ERG studies give us an idea that there is a decrease in the amplitude of the A and B wave in advanced stages of pathology as compared to non-transgenic littermates. Also, increased peak latency in photopic condition has been reported in animal models. Moreover, a significant reduction in mixed rod-cone responses, such as decreased A and B wave amplitudes in mice carrying the APOE allele, which is actually the most prevalent genetic risk factor for the late onset of Alzheimer's disease, has been also reported. Results from these studies emphasize the value of potential electroretinogram biomarkers in Alzheimer's disease. Now, we also have seen retinal measures from fundus images. And these are measures that comprise structural and vascular information of the vessels on the back of the eye. The vasculature network metrics that are characteristically used in the field are, for example, fractal dimension, which is a global summary measure of branching complexity of the retinal vascular tree, reflecting how truly the branching pattern fills 2D spaces. For example, larger values represent a more complex pattern, and as you can see in this figure here, you can notice that this is the retina with a high fractal dimension compared with another retinal image showing a lower fractal dimension. You can see that you can easily see the differences in the complexity of the vasculature. Another measure that has been extensively used is texture, and basically this is a measure that provides information about the spatial arrangement of color or intensities in an image of selected regions. For example, here you can see that I'm indicating two specific regions where we have exudates and where we also have retinal detachment. So you can see that the coloration and the granularity in these regions are different. Lacunarity is another measure that is used to identify heterogeneity or gap dispersion within an object. Particularly, an object with large gaps has a high lacunarity, while a low lacunarity is associated to homogeneous objects characterized by gaps that are all the same. We also have the artery vein diameter ratio, and this is a very well-known and well-used metric. For example, a lower ratio between the width of the arteries and veins is well established to be a predictive parameter of a stroke and other cardiovascular events in adults. And last but not least, we also have vessel and perfusion density, and also the blood flow velocity, which are vascular measures. Like, for example, if we use OCT angiography, we can identify an idea about the vessels in the back of the eye, as you can see in this figure here. Now, let me show you about our research approach and how we are managing seeing through the brain with our approach. The main goal in our study was to evaluate the feasibility of introducing a low-cost ophthalmic multimodal platform comprised by retinal imaging, electrophysiology, and visual function tests to better assess eye complications in elderly individuals that may have early Alzheimer's disease signs or be at the risk of cognitive impairment. Our objective was to find whether evaluating retinal alterations in individuals may add value to the early detection of Alzheimer's, and whether novel eye biomarkers for early detection of cognitive impairment in Alzheimer's disease could be introduced at a low cost. As you can see in these images, we are using fundus photography to acquire images of the back of the eye to check for features or abnormalities in the structure that may correlate with Alzheimer's. We are also investigating the circulation deficit in the vascular structure using a novel technology developed by VasoOptic Medical. We are also using electroretinography to measure the retinal function and other exams to test how well your eyes can detect changes in color and contrast. Before I move forward with more details of our study, I would like to mention that I eliminated some of the technical and complex information like mathematical derivations that are related to our research to make this presentation easily to understand for a general audience. However, more details of our studies can be found in our publications for anyone interested in more details. Now, in relation to study participants and procedures conducted under the study, we first identify individuals with cognitive impairment in a non-systematic fashion as they appear in the clinic, or identify them from a population attending adult daycare centers and community clinics using available medical records. Exclusion criteria were age under 55 years and the presence of any ophthalmic history, like for example, glaucoma or age-related macular degeneration. Also, patients were excluded if they had other brain pathologies like a stroke or Parkinson. I would like to mention that cognitive function assessment using the Montreal Cognitive Test, electroretinography using full field ERG with the LKC technology device, and color vision tests and advanced retinal imaging were used in the study. Provided the most recent results of the neuropsychological or screening test could be over 1 year for some of the patients with mild or moderate cognitive impairment, we conducted the Montreal Cognitive Assessment Test at the time of recruitment. To first eliminate any potential time gap between cognitive and ocular assessments, and second, to facilitate the standard screening metric for the cognition status that could be used in the analysis, independently of the subject's cognitive impairment causation or duration. Also, optic disc and macular center images were scrutinized for pathology, and subjects without any ocular history except for cataract surgery were included in the analysis. A smoking history and information on the presence of diabetes, hypertension, and cardiovascular disease were also obtained. As in many other studies, we conducted data analysis to detect significant trends and associations between the retinal and cognitive measures. As you can see in this figure, retinal fundus photographs were taken of each eye with a confocal laser ophthalmoscope, which is a non-mydriatic digital camera that suppresses scattered light and produces high contrast images, even in patients with cataracts. Also, evaluation of the bioelectrical activity of the retina was performed with the low-cost handheld alternative electroretinogram from LKC Technologies that can be used without pupil dilation and any eye contact. The electroretinogram measurements were acquired in both eyes in about 5 minutes without any eye contact. You can see in the figure that the sensor electrodes were placed below the lid margin, and these sensor electrodes are as user-friendly as a Band-Aid. We use the light-adapted ERG protocol in this particular study. Now, we also evaluated visual performance with a computer-based test known as the Rabin Cone Contrast Test. This is an easy-to-conduct exam to document contrast sensitivity and color vision assessment. So we conducted the test from the right eye to the left eye in a dark room, and it took about 5 minutes to complete per study subject. As you can see in this figure, the test displays color letters on a gray background, which are visible only to red, green, or blue cones. A staircase program, like a hearing test, adjusts color contrast up and down based on the observer's correct or incorrect responses to determine the lowest red, green, and blue cone contrast the patient can see. The red, green, and blue cone contrast test scores are expressed on a scale of 0 to 100 based on the number of letters identified correctly. Now, in relation to the image analysis, once vessels were segmented, the optic disc center image were partitioned into 9 equal regions observable in the full 45 degrees field of view, and the corresponding skeletons were obtained and used in the fractal analysis. I remember this is, the skeletons are the one used for calculating the fractal dimension, as I introduced earlier in the talk. Now, here are our results of the 69 participants. 32 of them had cognitive impairment, which is about 46%. As you can see, the table 1 is showing the study baseline characteristics. After applying all exclusion criteria, a total of 20 subjects with cognitive impairment and 19 cognitively healthy subjects were included in the final analysis. In contrast to cognitively healthy people, we found individuals with cognitive impairment that were characterized by lower fractal dimensions, so the vessel pattern in the back of the eye was less complex. We find that these individuals also have higher lacunarity values, so the vessel pattern in the back of the eye was more heterogeneous. The functional loss in color, we also found functional loss in color vision. Interestingly, most individuals with cognitive impairment had more green deficiency than red or blue deficiency in the cone contrast test. And also, we found a shorter amplitude and delayed electroretinogram response in these individuals with cognitive impairment. Regarding significant trends between cognitive scores and the retinal structural function, we also found fractal measures that were significantly associated with cognition scores, specifically with the Montreal Cognitive Assessment scores, and also correlated with electroretinogram metrics, and specifically with the implicit times of the individuals in the study. In general, we observed drusen-like regions in the peripheral retina, most of them in the superior quadrant, along with pigment dispersion, which was noted in subjects with cognitive impairment. The red arrows indicate the location of the drusen, and the white spots observed at extramacular locations. The areas enclosed here by the orange rectangles indicate the locations where pigment dispersion was observed. Two earlier studies that may add to this evidence have described the presence of amyloid beta in retinal drusen deposits, and also abundant amyloid beta pathology has been detected in Alzheimer's disease patients in the periphery of the superior quadrant. The logistic regression analyses were conducted only for the regional retinal sectors where the statistically significant parameters were correlated with the larger effect size. And this is about statistical analysis. So the optic disc region parameters were used in this analysis to identify the parameters or the combination of parameters with the greatest capacity to discriminate between cognitively healthy subjects and patients with cognitive impairment. As you can see in this figure, the receiver operating characteristic analysis is a useful tool for assessing the accuracy of model predictions. The bigger area under the curve, the better the prediction. While the table shows the results obtained for 5 models that use a different combination of parameters, the receiver operating characteristic graph shows only the results for only 3 of them. Note that the model number 5 is the one that offers the highest predictability for the cognitive impairment, with an area under the curve of 0.95. In this pilot study, a methodology was investigated to assess the feasibility of using a variety of eye biomarkers in the detection of Alzheimer's disease. I would like to mention that most of the studies trying to identify biomarkers for Alzheimer's are using an univariate approach. So in this particular pilot study, we tried to identify the potential of combining multimodal markers for the early detection of cognitive impairment. The majority of treatment for Alzheimer's disease has failed because intervention happens too late, because it is difficult to identify individuals with the disease in the early stages. Our preliminary results support the use of a multimodal diagnostic biomarker approach based on the retinal structure-function relationship, and has the advantage of a low-cost implementation that is non-invasive and user-friendly. However, while the full extent of our approach clinical applicability is provocative and is still to be determined, this study pointed us to explore more in detail the retinal biomarkers that reveal potential trends to identify individuals with cognitive impairment due to Alzheimer's disease by using a larger and more diverse sample size with a longitudinal study design. Last but not least, I would like to acknowledge our team and collaborators and all the research participants. And with all that said, I would like to thank you for your time. And I will be more than happy to address any questions that you may have.
Thank you so much, Dr. Cabrera DeBuc. I really enjoyed listening to your presentation, and I know I learned quite a bit that was new to me, and I'm sure our audience feels the same way. Again, I would like to ask the audience if at this time you have any questions, you submit them through the chat function. If we don't have time to address every question right now, we can get back to you offline once the webinar is done. But I see a few questions already coming in. So if you're okay, Doctor, we'll start with the first question.
Yeah.
First question is, I am wondering about electrophysiology as a biomarker in some psychiatric illness, as well, for example, major depressive disorder and bipolar. Do you have any thoughts on that?
Yes, electrophysiology, and in particular electroretinogram, has been used in psychiatric disorders. You know, actually, way before Alzheimer's disease, there are some review papers, interestingly, in the field that are commenting about the applications of ERG or electroretinogram for bipolar disorders and the other diseases that any psychiatric doctor may be probably targeting.
Thank you.
You're
The
welcome.
next question is, aiming on easily accessible tools to make them basic test, which of the 3 devices you presented would you recommend?
Okay, so your question is basically, what is my preference, using a fundoscopy, which is using a retinal camera, an electroretinogram, or contrast sensitivity test, correct? So
Yes.
That's your question. Just keep in mind that in my particular study, I'm fusing the information from the structure and the function, because the more information you can obtain from the eye that may be related to what is happening in the brain, the better will be the predictive biomarkers. So I prefer to use multivariate biomarkers. In terms of user friendliness, I think that the electroretinogram device from LKC Technologies, which is a handheld device, is very convenient. It's very user friendly to use. In 5 minutes, you can scan both eyes, and the patient's tolerance is really good. It's not uncomfortable at all. And one of the key advantages and one of the main decisions I took when I decided to use this device is because it's insensitive to moderate opacities in the eye. So for example, if you have to take care of patients, like for example, all the older population or the elderly, you will see that they may have cataract already. So you want to use a technology that is not going to be impacted by these particular media opacities, which are very common in the old people, you know. But yeah, usually in terms of research, I prefer to fuse information using the multimodal or the multivariate approach, because the predictive biomarker should provide more information when you obtain information from different features in the eye. But in terms of user friendliness, I like the handheld device. It's very user friendly. And there are handheld cameras, of course, for the same objective, but the device that I'm using is a tabletop camera right now.
Thank you.
Yeah.
The next question that I see has to do with getting slides of the presentation. And actually, I just wanted to note for everyone, whoever is registered will be receiving a recording of this webinar after the fact. So that will be coming out to you shortly. The next question is, how does refractive error affect the equipment to measure the ERG?
Well, when we measure the ERG, actually, if the patient, for example, is using glasses, we ask the patient to remove the glasses, okay? Because this is just a light that we are flashing into the eye. So you don't want to have another mirror of the glass that is going to be reflecting back the light that you want to put into the eye. So that's one of the things that if the patient has a refractive error, it has to remove the eyes, and then you have to take the measurements without the glasses. So that's the way it is.
Great, thank you.
Yeah, well.
Okay, the next question says, you mentioned that you wanted to be able to predict Alzheimer's disease with tests that are easy and quick to perform. You mentioned that the RETeval test took about 5 minutes. How long did the other measurements take?
Well, imagine that the confirmatory biomarker testings that are currently used are positron emission tomography, which is a neuroimaging modality. Keep in mind that will require probably 45 1-hour minutes after you are injected with the radioisotope. You know, it's actually quite well invasive, so it takes time. It takes time to even schedule to have a positron emission tomography scan. It's not as easy as scanning the eye that may take 5 minutes. The other confirmatory biomarker test is the cerebrospinal fluid test. So that's even more inconvenient if you consider, you know, that you have to actually deal with the patient in a very uncomfortable manner with this particular test. It's not like the eye, it's really non-invasive. It's a non-contact tool. It's very user-friendly. 5 minutes, so that's a huge difference there. And in my experience, the patients really are happy with this technology.
That's nice to hear. Thank you. The next question is, what is the consequence of reduced vascularity?
Well, just keep in mind what is happening is that you may have a circulation deficit, all right? So you may have, for example, that you probably have gaps in the structure. There are things that are actually not like when you are a young person. And this is not only happening in the eye, this happens with many other organs, all right? So you may have degeneration going on. So that's pretty much showing probably circulation deficit on the back of the eye, or a structure that may not be working as before. Could be affecting, for example, the neurovascular coupling, you know, how much those vessels are being perfused and how much they may be feeding in terms of nutrients, you know, the structure or the back of the eye, the tissue, the retinal tissue.
Okay, thank you. Next question is, in your opinion, if you were trying to follow a diabetic patient who also has Alzheimer's disease, would the ERG be confounded?
This is a good question. And the good thing is that actually patients with diabetes, you know, have to follow the risk of Alzheimer's disease, all right? And when you see, you can pretty much conditions in the back of the eye in a diabetic subject that may be paralleling what is happening in Alzheimer's disease subjects. So things can be confounded. And this is one of the key issues that I'm trying to address now, because when the people get old, they can have hypertension, they can have diabetes, they may have cognitive problems, they may have other chronic conditions that may be affecting actually the back of the eye, the structure, the function. So exactly we need to find a differential diagnosis. Is what I'm observing on the back of the eye because of the Alzheimer, or because of the diabetes, or because of the hypertension? And this will require actually to work towards differential diagnosis, that we can scan patients that may have Alzheimer and not diabetes, for example, or Alzheimer's and not hypertension, and try to see if there is actual differences in the patterns from the back of the eye in terms of the response to the electrical stimulus using the flash ERG with the LKC technology device. So this is a long way to explore, and differential diagnosis is a very attractive solution. It has to be done. We need to actually keep in mind all the confounders to actually get better predictors for the particular disease, independently of Alzheimer's, diabetes, or whatever other type of condition.
Thank you. There's a few questions that probably I can combine here. Multiple people are asking if ERG biomarkers can differentiate between Alzheimer's and other types of dementia like alcoholic dementia or ischemic dementia?
That's another difficult question to answer. As I said, we have vascular dementia, we have Lewy bodies dementia, we have mild cognitive impairment. So we have different stages. Alzheimer's disease as a continuum, you know, so there are different stages on top of that, and there are different causes of dementia. Dementia is a general term. So there are many, many different types of dementia: Alzheimer's, Lewy bodies dementia, the vascular dementia, and so forth. I think that there are about 13 types of dementia. So this is one of the things that many research groups are focusing, that we need to actually find differential diagnosis. We need to find, for example, changes in the structure, assuming that is the eye a good biomarker for this particular condition. Changes in the structure that give us actually a specific differential points that allow us to predict better what may be the causation in the problem.
Thank you.
But definitely, you know, it is possible. It is possible because depending on the stimulus that you can use with the electroretinogram, for example, you may be getting response from the ganglion cells or from the amacrine cells. It depends on the type of protocol that you're using to acquire the bioelectrical response of the retina. So it is just the way it should be done. There's a proper protocol, proper differentiation, proper selection of patients in which you can control your confounders, of course, and see what is the response, and if there is any difference in the response that may be used as a differential marker.
Thank you.
You're welcome.
The next question, well, it says, thank you for an amazing conference. I would like to know the Model 5 in ROC analysis, including which exam combinations.
I did notice a ROC analysis. What was the analysis mentioned?
Model 5 in ROC analysis.
Okay, the ROC analysis that we conducted?
Yes, including which exam combinations.
Oh, okay, I got you. Yeah, the ROC analysis, keep in mind that we actually tested different models combining multiple parameters. For example, we have one model that only use the fractal dimension parameters. Another model that uses the fractal dimension parameter and the spectrum singularity parameters. And I'm going to enter now in the mathematical definitions that I wanted to avoid from the very beginning. But then I'm sure that this person asking this question might be very familiar. So you have to combine fractal dimension, spectrum singularity parameters, then you enter into lacunarity, and then you have the implicit time parameters. You see, so we have about 5 models in which we were actually adding and removing parameters and trying to see which one was actually giving us a much better or bigger area under the curve, which is the ROC analysis. The bigger the area, the better the prediction of the biomarkers. And we found that the more parameters, of course, that you add from structure and the function give us a much better predictability. So we have 3 models plot on the figure that I show you, which were the fractal dimensions. And if I recall properly, the second model was combining fractal dimension and singularity spectrums, and the last one was combining all of them with the lacunarity and implicit time, which is model 5, I think.
Thank you.
You're welcome.
Next question is, which protocols from RETeval did you choose to analyze Alzheimer's disease?
Well, in particular for this study, we use the light adapted, which is the flicker protocol that is in the device. And we, at the beginning, when we start recruiting this population, we didn't use a photopic negative response, but in our latest patients, we're having this. There is a combined protocol in which you can go through the light adapted, and then you finish with the protocol for the photopic negative response activity. That is a
Great.
standard light adapted protocol, yeah, the flicker response.
Okay, the next question says, if I understood you correctly, the electrophysiological electrophysiological biomarker was reduced amplitude and delayed flicker ERG. Did you find any other biomarkers such as photopic negative response or the cone on-off response?
No. And in this particular study, this was a pilot study that when we started, we didn't have the photopic negative response in the device yet integrated. So we only used the light adapted. So it was basically the amplitudes of the A and the B wave and the implicit time. Those were the only parameters that we were able to analyze with this protocol.
Okay, the next question is, do you have a theory why M cones seem to be more affected in Alzheimer's disease?
The M cones? I think that probably has to do with the influence on the ganglion cells, influence from the inner retina, but everything that is related to Alzheimer is still under exploration. You know, this is pretty much still an area that is on the research.
The next question is, are any similar findings found in Parkinson's disease?
With the ERG?
Yes.
Yes, in Parkinson's disease, we have not used electroretinography in Parkinson's disease, and we have not explored too much with even with fundus photography this particular disease. So I cannot really comment on Parkinson's disease, but I know there are reviews in the literature, mostly analyzing with different technologies, you know, but with electroretinography, I'm not quite familiar with the Parkinson's disease at all.
Okay, the next question says, are you doing scotopic and photopic testing on these patients?
Yeah, we tried the scotopic at the very beginning, but what's happening is that we realized we use patients with mild cognitive impairment and also patients that had some kind of moderate cognitive impairment, and they were not too happy, you know, staying in a room 20 minutes in a dark room. So even though we have data from the very beginning, we have both photopic and scotopic measurements from pretty much almost every patient, but we decided not to follow with that protocol because some of them were really not too happy to complete the measurements, you know, mostly the ones with moderate, you know. But we found interesting trends, reduced amplitude. We found very good interesting trends with the scotopic response, but it was not user friendly. One of the reasons I decided to use first the LKC technologies device was because it's very user friendly, can be deployed anywhere. You don't need to dilate the pupils. You don't need eye contact. So it was very user friendly. You can even have somebody that is not an ophthalmic specialist or technician to use the device. So that was my main reason. I wanted to use a technology that can be deployed anywhere. So provided that a biomarker could be obtained, then this is something that can be integrated easily in any clinical setting.
Thank you.
You're welcome.
The next question, have you been able to use OCT and electrophysiology to determine dementia, such as loss or thinning of certain layers in their retina?
Yes, no, I'm not using OCT, although OCT could be one interesting technology to use. And actually, there are many groups trying to use OCT, not only from the neurodegenerative point of view, in which there are reports in which the retinal nerve fiber layer and the ganglion cell complex are actually getting thinner in these patients because of the neurodegeneration. But also the OCT with the angiography component is also producing interesting trends with the vasculature or the perfusion information on the back of the eye. But I'm particularly not using OCT because my interest is to use a low-cost approach that can be deployed and integrated easily if the biomarker is good enough. But OCT is a good technology. It's a very good technology, and many groups are reporting good trends. Some of them contradictory, but yes, I think that we need to standardize the measurements so we can do better. Yeah.
The next question for you is, do you think we could use full field ERG to look for a response to treatment?
Response to treatment. Well, keep in mind one thing that is also a concern when you measure the full field response, you are getting a sum of the response of what may be happening in the back of the eye. Okay, so it's not like for example when you use multifocal ERG or pattern ERG, so you get a global response of what may be happening on the back of the eye. In terms of trying to see the response to treatment, I think that has to be quite well controlled to figure it out if this is something that actually is going to help, you know. But I'm not doing anything with treatment, okay? I'm just on the upfront in which I want to determine first if we can detect the problem. But treatment to response, I think, is pretty valid, depending on exactly what type of treatment you want to introduce, and see what type of response may be expected of that treatment, what type of retinal cells, for example, you may be targeting with a particular therapy.
Okay, thank you. The next question has to do with, it says, what percentage of the subjects did you get measurable results from? You mentioned cataracts could affect fundus imaging.
What percentage?
What percentage of your subjects did you get measurable results from?
In most of them, actually, because most of the patients, we were very careful, not because of the electroretinogram device, because we know that it can cope with that particular condition, but even for the fundoscopy imaging, we try to not get advanced cataracts or nothing media opacity, although we use, you know, the confocal machine that was very friendly also with those type of media opacity. So I didn't have much problem at all. First, I try not to have advanced cataracts, of course, but the devices that we use were very friendly in relation to cataracts, the cataract present in the eye.
Okay, the next question is, one of your slides showed that the contrast sensitivity of VEG is increased from Alzheimer's disease patients. Any explanation?
Yes, the contrast sensitivity, well, as I mentioned, we have patients that do have the red, the green, and the blue, and there were actually more deficit in the green stimulus for them in this particular group of patients. Keep in mind that we have patients with mild cognitive impairment and with also moderate. So our study group was actually mixed. It was not that they were only at the early stage, but also we have a mixed population that we were able to put together because of the amount of confounders. We couldn't do better for that particular pilot study. But essentially, the contrast sensitivity is one of the deficits or visual deficits that have been reported in the literature, and that is affecting most of the patients with Alzheimer's.
Okay, thank you. So it looks like we are quickly approaching the end of our webinar time. We have quite a few questions left. I want to assure everyone that we will get in touch with you offline and respond back to all of these questions. And I particularly thank a lot of our audience that's in Europe right now in the middle of the night submitting questions. We will get back to you. I will do one last question, Dr. Cabrera DeBuc, because I think this is a great one to end on. What is next for your research? Where do you see your research going next?
Yes, the next steps that we are doing right now, we want to recruit patients in which we can control better the confounders. So we are working towards finding differential diagnosis. So we want to characterize, for example, subjects with the same age, for example, and we also want to stratify them by the gender, because you know that women are more affected by Alzheimer's than men. And we would like to see the trends in a more stratified manner with the methodology that we are following. So that's the next step. We are moving forward to recruit patients and try to stratify better the data and try to see if we can still obtain the same data that we obtain even when we have a population of subjects this time that was not as expected, or at least as good as we wanted, you know.
Thank you. And we all look forward to seeing where that research takes you, and we'll be happy to have you back to talk about it.
You're welcome.
I just wanted to take a minute to let everyone know in the audience that we will be wrapping up now. We would be happy to hear from you, any input that you have. There will be a questionnaire at the end of this webinar, so any input that you have back for us, or if you have additional questions, please feel free to leave them there. We are having a few more presenters in this webinar series, and we would be happy for you to join us for them as well. You can register online through our website, LinkedIn account, or Facebook. And please be sure to share them with your colleagues or students that may be interested as well. The last thing I wanted to mention was if you are interested in learning more about LKC's ERG devices, you can visit our website or contact us directly at info@lkc.com. And I wanted to thank you again, Dr. Cabrera DeBuc, for joining us and for your wonderful presentation. We really enjoyed having you, and we hope everyone in the audience enjoyed it as well, and everyone will join us again in about 2 weeks.
Thank you. It's a pleasure.
Thank you.
Good night, everybody.